Hello, my name is Ernie Guzman, and I am the Senior Manager of Technical Support here at Twist Bioscience. And I'd like to welcome all of you to today's presentation entitled Targeted Tools for SARS-CoV-2 Research. It looks like we have a number of people joining us, people from Boston, upstate New York. Cool, upstate New York. I went to grad school there. We have some visitors from Brazil, Spain, bienvenidos, Belgium, Rome, everywhere. So that's great. I would like to say thank you and welcome to everyone. Today, we will have two speakers who will share how Twist has responded to the COVID-19 pandemic by developing products used in disease testing, surveillance, vaccine development, and therapeutic antibody discoveries. Just a couple of quick housekeeping rules to cover. First, all lines will be muted during the webinar. This is to reduce any distractions during the presentations. Secondly, we will have a Q&A session at the end. Please submit your questions in the Q&A box. We will answer your questions after the presentations are complete. And finally, following the presentation, there will be a short 1-minute survey that we would really appreciate you taking. Your feedback will help us improve future webinars. Okay, so let's get started. Our first speaker is Venkatesh Moktali. Venkatesh is a Senior Product Manager at Twist Bioscience, managing our targeted enrichment panels and software portfolio. He has extensive experience working in the genomics industry and a doctoral degree studying the evolution of plant and human fungal pathogens. Our second presenter is Mark Consugar. Mark is a master-level scientist with more than 20 years of academic, clinical, diagnostic, and translational research experience in inherited and somatic genetics. Mark also has more than 10 years of NGS experience with whole genome sequencing, whole exome sequencing, target enrichment, and transcriptomics. He has been with Twist for 3 years as an NGS field application scientist and was recently promoted to field application manager. So with that, Venkatesh, the floor is yours.
Thank you, Ernie, for the kind introduction. Mark, if you could skip to the next slide, please. Perfect. Thank you, everyone, for joining us today. What I want to do today is start with a very brief introduction to Twist and what we do at Twist. Then I'll go over an overview of the SARS-CoV-2 products that Twist has launched over the last month and a half. And then I'll hand it over to Mark for a technical deep dive on our NGS panel that we've launched. Next slide, please. Okay, so let me dive straight in. So Twist was founded in 2013 on a platform technology that enabled us to synthesize DNA at massive scale with high precision on a silicon platform. So this silicon platform is what separates us from everyone else in its ability to scale the chemistry to very small reactions using the silicon-based platform here. And what has happened due to that miniaturization is that we are able to provide really high throughput while maintaining quality as well as speed at scale. Next slide. So using this platform for our customers, we provide a variety of products. These include genes, oligo pools, variant libraries, NGS panels, as well as data storage. And we are working really hard in all of these areas to provide you with the latest products. Next slide. So using this platform in the last month, we've been working hard to come up with mainly two specific products to deal with the coronavirus epidemic. These are namely the synthetic viral RNA controls. And then we also launched along with that a SARS-CoV-2 Research Panel for NGS Target Enrichment. You can find all of these product details on our website today. Next slide, please. Let me tell you a little bit more about the synthetic viral RNA controls. So we started making controls for 2 strains. The control 1 originates from Wuhan in China, while the second one originated in Australia. These controls consist mainly of 6 non-overlapping 5 kb fragments covering the entire viral genome. In fact, about 99.9% of the viral genome, and the remaining small bit is constituted by a very short poly-A tail that we exclude from what you get. We provide these controls in 100 microliter tubes with a concentration of approximately 1 million copies per microliter. Currently, what we do is, because these are controls made up of synthetic non-overlapping viral RNA, they are suitable for BSL-1 facilities. So you don't have to have any specific setup at your lab. You can use it straight away in your existing setup, and it's completely safe to use. As I said before, you can order this product through our e-commerce platform, through our website, and we can go to the link twistbioscience.com/shop, and you should be able to add all these products to the cart and check out in a very simple to use workflow. Next slide, please. Since we started working on the synthetic viral RNA controls, there have been about 100 different variants of this virus in public databases. We've already started working on launching additional synthetic controls, and some of them should come out over the next week and a half. If there's a strain of your interest that you would like a control synthesized for, reach out to us, and we can make them available to you in approximately 2 weeks. Next slide, please. I'll switch gear quickly to the research panel here, the NGS panel, and talk to you a little bit about what this panel looks like. At Twist, we've launched several panels in the last couple of years with excellent performance, and this is no different. This also follows the same trajectory of the other panels we've launched. It includes approximately about 1,000 probes that target the full SARS-CoV-2 reference genome. The panel goes through the same rigor, as I mentioned, compared to all of our other gene panel products, where we NGS QC the final probe library. Today, it is available in 3 reaction sizes. There is a 2 reaction size. There's 12 and 96 for different kinds of requirements that you would have. Again, these are all orderable through our e-commerce platform, and you can buy them today through that easy workflow that I mentioned. With that, I'll end my part of the talk and open it up for a more exciting session with more in-depth details from Mark. And before I do that, I'd like your opinion through a poll question that is coming up.
Okay, so the poll question, what would you use the SARS-CoV-2 synthetic RNA controls for? The two options are quantitative limit of detection measurement assay or a qualitative viral presence or absence report. And taking a look at the results, it's interesting, actually. It looks like almost a perfectly even split, a 50-50 split. So that's really interesting. Okay, so looks like the poll is closed, and it looks like a pretty even, it's a 52-48 split with quantitative limit of detection measurement just eking it out by 4 percentage points. So Mark, I think we're ready now for your part of the presentation. Take it away.
Very good. Thank you very much, Ernie. I appreciate you handling the poll question. Now I'm going to move on to the SARS-CoV-2 Target Enrichment panel data analysis. So with that, just to start, I just want to echo Venkatesh's information with regard to our SARS-CoV-2 homepage. This is where our customers can find anything and everything which is SARS-CoV-2, including our central repository of literature, our products, and also CDC and FDA information. Before I get into the target enrichment research panel that we've developed for SARS-CoV-2, I would like to give a little bit of background as to the details as to what went into the development of this particular panel. So SARS-CoV-2 shares homology with a number of other respiratory viruses. So in order to come up with a panel that would be as specific to SARS-CoV-2 as possible, we needed to look at all of the, let's call them nearest cousins, brothers, sister viruses that would be relative to SARS-CoV-2, so that we would have a better understanding of where the probe placement would occur, what would be specific probes versus homologous probes, and how best to understand the need for complete versus partial genome coverage in next generation sequencing. So as you can see here, when our informaticians looked at SARS-CoV-2 versus SARS, MERS, and 4 other very common coronaviruses associated with respiratory disease, you see a range of anywhere from 50 to 78% homology in the genome sequence. When you dive a little bit deeper into that, those probe sequences and the corresponding genome that we are trying to capture for SARS-CoV-2, you can see that the probe sequences relative to the coronaviruses match up pretty much in line with the homology mapping that I had shown on the previous slide, where approximately 80 to 90% sequence homology is observed across a median distribution for SARS-CoV-2 or for SARS versus MERS versus other coronaviruses, which is shown here in the upper left-hand distribution plot. However, when we take a deeper dive into probe composition and sequence homology, of the 994 120 base pair probes that we have in our SARS-CoV-2 panel, we have identified that approximately 15% of them, or 159 probes, have less than 70% sequence homology, which means that they are going to selectively capture fragments that correspond to SARS-CoV-2 selectively over any other homologous region in a sister or cousin strand of this particular virus. That data is shown here on the bottom, where we can see the Wuhan strain of the genome presented here in the top plot of the IGV trace. And in the bottom, you can see these unique regions, which are pretty much spanning across the complete 30 kb region, but seem to have density in both the 5' and the 3' ends. What we decided to do, though, was to go a little bit deeper in doing what I like to call SARS-CoV-2 genome fingerprinting, which is to say, rather than thinking about sequence similarity or contiguous sequence similarity, what is the single base pair differentiation of SARS-CoV-2 versus SARS in specific stretches of DNA? In essence, are there regions throughout the genome, or is there, in essence, all throughout the genome, single nucleotide mismatches that are scattered throughout? So that even though we're looking in the previous slide with our probes being 70% or less homologous based on stretches of sequence similarity, what happens if you just look at it on a single nucleotide base resolution? And what we have found is that if you break up the SARS-CoV-2 genome into 299 100 base pair segments, almost 99% of the genome has at least 5 nucleotides which mismatch in each one of those 100 base pair regions. So in truth, there are a lot of single nucleotide positions throughout the genome that you can utilize to identify or fingerprint SARS-CoV-2 versus SARS or another coronavirus, even if you don't have 100% genome coverage of the particular sample or of the particular virus that you're looking for in this particular case. So I believe, as I show data in the coming slides, that this is going to have some resolution into how target enrichment can be used as both a supplementary and as a complementary method for qPCR. With that, I'm going to open this up to our next poll question. Ernie, take it away.
Okay, the poll question. The poll question is, what would you use? No, that's the first poll question. Second poll question. Here we go. Yeah, so what would you use the SARS-CoV-2 NGS panels primarily for? It's multiple choice. Viral detection, surveillance analysis, and viral evolution and strain typing. Just give it a couple of seconds here. Okay, so it looks like the vast majority, or a fair majority here, is viral evolution and strain typing, with 71% of respondents saying that that's what they would use it for. surveillance analysis coming in second, and finally viral detection third. So yeah, viral evolution. Thanks, Mark. Take it away.
Right. So moving on, what I'd like to do is start diving into the methodology that Twist has developed for generating libraries from samples to generate double-stranded DNA for target enrichment and then sequencing. So here is our workflow in a nutshell, spaced out in 4 distinct regions, which is generation of double-stranded DNA from the virus, which can then be used to generate a library, which would then move on to hybridization and target enrichment, and finally to NGS sequencing. So to generate the cDNA from the single-strand RNA of SARS-CoV-2, what we do is we take a protoscript 2 first-stranded cDNA synthesis kit from NEB and generate the cDNA. So in this case, you now have a cDNA RNA hybrid. We then take, and that is done by using the random primer 6 also from NEB. Secondly, we will take the NEB Next Ultra 2 non-directional RNA second strand kit, and we will generate double-stranded DNA from the cDNA single-strand RNA hybrid, which was in the first step. So at this point, we're taking single-strand DNA through to double-stranded, or single-stranded RNA to double-stranded DNA. I would like to comment here, because I will make mention of this later. This methodology was developed so that we could simultaneously isolate and analyze both DNA and RNA-based viruses. In this particular case with SARS-CoV-2, it's a single-strand RNA virus, but with additional product offerings that we will be making in Q2, Q3, the advantage of having a library prep kit that can do both DNA and RNA virus is going to be advantageous. We are going to then move on from having double-stranded DNA into the double-stranded library construction process. This is where you would end up taking your sample and you will break it up into smaller fragments, and you can use that either using an enzymatic fragmentation kit or mechanical fragmentation. Both kits are available through Twist Bioscience's sales specialists. And in this case, you would also use our universal dual indexed adapters. We currently have 400 10 base pair dual indexed adapters that we can utilize. However, that number will be increasing throughout Q2 and Q3. So the number of available indexes, and as a result, the number of available samples you can analyze simultaneously, will become increasingly more available to you as the year progresses. Now, we would then move on to hybridization. You would end up utilizing, in this particular case, SARS-CoV-2 target enrichment probes. These are the 994 probes that I had made mention to a few slides earlier. However, you can augment these probes with maybe other viral strains or other targets that you might have of interest. So this does not necessarily need to be locked in place. You can customize this panel, but in this particular instance, we're focusing primarily on SARS-CoV-2. You can then perform either standard hybridization, which is our conventional 16-hour hybridization, or pursue fast hybridization methodologies, which can do anywhere from 15 minutes to 4-hour hybridization. I'll show some data on this a little bit later. And then finally, capture and purify those enriched targets using the streptavidin beads and standard purification beads. Finally, you would end up doing next generation sequencing. Our current suggestion is to do a 2x76 base pair read. Based on the data that we've seen, both internally and externally, anywhere between 25,000 and 8 million reads may be required per sample based on viral loading and the amount of samples you want to run in any given particular sequencing run and or the platform that you're using to sequence. But it does seem to be that there is a sweet spot of about between 250,000 and 1 million bases or 1 million reads that will sufficiently cover the genome at a number of viral copies. So with that, I'd like to get into our next data set. So I'd like to show here our SARS-CoV-2 standard hybridization at 1x genome coverage or greater in 2 different formats. So the first format is percent genome coverage versus viral copies. And you can see here that I'm showing that in each bar graph plot, you have 25,000, 100,000, 250,000, up to 8 million reads at specific viral copies on the x-axis, which goes 1 million, 1,000, 10, and then the negative. What should be obvious from this is that at 1,000 and 1 million copies, we're getting complete coverage of the genome regardless of the amount of reads that we assign to the sample. However, at around 10 copies, we're starting to see that you will get erosion of genome coverage dependent upon the amount of reads that you are going to assign to the sample in question. And the range goes from anywhere from approximately 91% at 8 million reads, all the way down to approximately 18% at 25,000 reads. But if we're going to consider, so this is where in these lower viral copy numbers, the idea of a viral genome finger typing philosophy may come into play, where rather than having to have complete coverage, if all you're looking for is a plus or minus or binary result, that you will most likely have sufficient coverage of the genome to use those SNP fingerprints to assess if you have SARS-CoV-2 versus another coronavirus. An alternative way of looking at this, which I thought is very handy, is also the percent genome coverage versus the number of reads that you assign, where the bar graphs now represent whether you have negative controls or increasing viral copies. So in both instances, you're going to see that we end up having complete coverage at 1,000 or 1 million viral copies, but you will start to see less coverage at the lower copies, particularly 10, and it will start to erode as you go with less and less reads assigned. To summarize, for our standard hybridization, I'd like to provide this plot. I think this is a nice cross-correlation between viral copy and read number. And here you can better see the depiction of the advantage of using possibly the fingerprinting philosophy if what you're trying to do is maybe have more incomplete coverage of the genome, but want to increase the amount of samples. So for example, at 10 copies, if what you're looking for is just a binary result, you may be able to use anywhere between 100 and 250,000 reads to get sufficient coverage of the genome to make a positive call. However, if you're dealing with a large viral load, you may not need 1 million reads to be assigned per sample, because 1 million reads will give you 99.9%, but so will 25,000 reads. So in this case, if you do have an increased number of viral copies, you could assign less reads and just run more samples. In my next slide, I would like to show the SARS-CoV-2 coverage representation on IGV with relative to copies and genome position. As you can see here, with the decreasing number of viral copies from the top down across the Wuhan strain of the genome, we have complete coverage to pretty much 1,000 genome or viral equivalents. And then as you get to 10 copies, you're starting to see that we're going to have more and more gaps that are going to pop up in between sufficiently covered regions of the genome. And then when you're getting down to what we are showing here as 1 copy and then down to a negative, you start to almost see nothing available at all. So there is going to be shown a decreasing amount of coverage versus viral copies, which should be expected. However, at what seems to be epidemiologically relevant viral copies, i.e. approximately 1,000 and greater, we're getting very, very good coverage of SARS-CoV-2, in this particular case, the Wuhan strain, for the complete genome. So what I'd like to do is I'd like to maybe whet your appetite a little bit with showing some very preliminary fast hybridization data that we've been able to generate internally for utilization with SARS-CoV-2 NGS panel. Again, this is somewhat of a busy slide, but I think it's still going to be very informative for all of for this audience. Is that on the left-hand graph, I'm showing percent of genome coverage at 1x versus viral copies using either a 15-minute or 3-hour fast hybridization method. What you can see from this particular plot is that either at 15 minutes or at 3 hours of hybridization, we are getting almost complete coverage at 1x of 1,000 or 1 million copies of the virus. Excuse me. However, as you start to go below 1,000 copies, there is variability that starts to creep in with the fast hybridization method, such that it's getting to become a little bit more stochastic in the coverage of the genome. This is not unbeknownst to us, we're aware of it, but this also lends itself to the fact that the fast hybridization methodology, in particular, the length of the time for hybridization, the temperature for the hybridization, as well as the wash one temperature to wash off any non-specific targets relative to the SARS-CoV-2 genome, has to be finer tuned relative to a 16-hour standard hybridization method. I'd also like to comment at this point that because SARS-CoV-2 is approximately 62% AT rich, very stringent or very high temperature washes or hybridizations can start to induce some melting of probe versus target regions, and as a result, more finer tuning of the ideal temperatures for optimal performance will have to be vetted, in particular at these lower copy numbers. And then conversely, I'm also showing this plot again as reads assigned per time relative to percent coverage at the -10, 1,000, and 1 million copies. So one other time, so to show this in another vein, once again in a tabulated format, we can see that at 1,000 and 1 million copies, we have very stable, very complete coverage of the SARS-CoV-2 genome at either of the temperature or the times, the hybridization times that we had considered. However, what you do see at 10 copies is that there is some inconsistency, stochasticity, which is coming in. And this is indicative of the idea that at these lower copies, we still have some work to do with fine-tuning this methodology. And I would like to highlight that with 3 hours, in most cases, people would expect a more stable or a more complete coverage of the genome relative to at 15 minutes. But that's the inverse of what we're seeing. And once again, that's not a surprise here. It's just reiterating the fact that there's a little bit more work that we need to do, especially when the number of viral copies or the viral load is going to be impacted to a larger extent by the background, which is induced by the human. And ensuring that the limited copies that we have present in the sample is sufficiently captured because of the genomic composition of the genome, in particular that 62% AT richness. So in summary, from both Venki's and my presentation, I would like to make the following comments. Twist Bioscience has synthesized and developed 2 SARS-CoV-2 controls. The 2 current controls that we have available to us right now are the Wuhan and Australian strains. However, as Venki had very appropriately made mention of, is that additional synthetic controls can and will become available as new SARS-CoV-2 strains become more prevalent. And this could be more prevalent in general or more prevalent in specific regions of the world. Twist has also developed this SARS-CoV-2 Target Enrichment Panel, which I had just previously shown data for. This is a fantastic methodology for both surveillance and monitoring of SARS-CoV-2 genomes, whether it's the 2 that we currently have controls for, or those novel genomes that we're starting to see crop up due to the natural mutation rate of a viral strain. I think of this as not only a complementary assay to qPCR that was primarily going to be dependent upon viral load, the required genome coverage of a particular laboratory or assay, or the number of samples that you might want to be running in that particular laboratory or on that particular run, but that it could also be considered as a supplementary assay to qPCR. And this is particular in regions of high demand, in essence, where there's not sufficient testing in hotspot regions for the qPCR, so we need an alternative or an auxiliary test to be able to give us not only a monitoring and surveillance assay, but also a binary assay of whether you have the presence of SARS-CoV-2 and a general idea of how many viral copies you do have, but also in low availability or low accessibility regions, such as Latin America and South America, where the qPCR reagents may be either hard to get or the product chain is a little bit broken right now because of the high demand in other parts of the country or other parts of the world. We have also presented both standard and fast methods that we've evaluated internally. Our standard method is our method of choice at this point because it is providing us the most reproducible and stable results across the logs of viral copies that we've seen using our controls. However, we and others in the field have been using our fast method as a means of getting sample to sequencing result very quickly, especially in those cases where you know that you have a higher viral titer in a sample, and you know that you're going to have probably far more availability of the virus to result in complete genome coverage. And finally, just to highlight once again that our library construction workflow is compatible with both DNA and RNA viruses. And that, I think, is a very nice segue for some food for thought as we move through the COVID-19 pandemic, but also into the next phases of research, and in particular, infectious disease research throughout the year. First, the fact that a Twist comprehensive respiratory panel is currently in development and evaluation. This is slated to be released in calendar Q2, the Q2 quarter. This will include both RNA and DNA viruses and will be optimized for both standard and fast hybridization protocols. So once again, having this universal protocol that we've developed in conjunction with the NEB reagents is allowing us to be able to simultaneously analyze a diverse number of viral vectors that are causing respiratory virus, whether they're DNA or RNA centric. We're also in the process of developing and releasing a Panviral 2.0 panel. This is slated for a Q2, Q3 release. Again, this will include both RNA and DNA viruses, including those viruses that we have in our respiratory panel, such as respiratory viruses, flu viruses, and SARS-CoV-2. Again, these will be optimized for standard and fast hybridization protocols. And finally, before I take questions, I would like to comment that SARS-CoV-2 continues to mutate. And so surveillance and monitoring assays are going to need to be able to accommodate sequencing mismatches that are going to logically and naturally become more and more prevalent in the base genome sequence of these SARS-CoV-2 strains that we started with, whether they happen to be the Wuhan strain or the Australian strain. But also, the assays should be able to easily accommodate customization and the addition of new content as we wish to expand either unique regions, whether they happen to be indels or additional content for the SARS-CoV-2 genomes, whether they be mutated or a completely new strain. Or, for example, if we want to try and survey and monitor the possibility of having co-infections due to two different types of strains of virus which may be subject to a patient, or accurately identifying and qualifying a treatment course that is specific to the virus that we're looking at, and not assuming that it's always going to be SARS-CoV-2, but if we do not see it, how else do we treat said patient? So I firmly believe that we need to be in a position where not only do we deal with the pandemic at hand, but be able to accurately and reliably be able to deal what this pandemic will teach us about infectious disease and the next possible infectious cases that we'll see in the near future. So with that, I would like to open this up for any questions that our audience may have.
Okay, well, thanks, Mark. I'd like to actually thank both of our speakers, Venkatesh and Mark. There are a couple of questions that people have. One of the questions, I think, maybe for Venki here. In surveillance mode, can your pipeline call COVID as well as other COVID variants in multiple samples? And along with that, what about adding flu and a few other clinically relevant viruses?
Great question. I think Mark alluded to some of this in his last slide. Of course, you can. The answer is yes. You can call other SARS-CoV-2 variants through our panel, as well as going as far as other respiratory viruses. There is a fair bit of sequence conservation between these viruses. But however, of course, we don't. What we would recommend is that you want to take a look at all sorts of variation within the SARS-CoV-2 virus itself. As Mark mentioned, we are also in the process of coming up with a respiratory panel that actually expands beyond the coronaviruses into other respiratory viruses, and we are working on it as of now, and we'll come out with it probably closer to the end of this month. So stay tuned for that.
Okay, thank you so much. There's a question from one of the attendees. Mark, can you just sort of address real quick or re-answer the question about how big the probes are?
Yes, our SARS-CoV target enrichment probes are 120 base pairs in length. That is the standard length of all Twist Bioscience probes. Okay,
and I think a companion question to that is, deletions and mutations may hamper binding of the viral probes. So how do we solve that, or what's the effect of how are we solving that issue?
So the probes are sufficiently tiled across the genome. So depending upon what the length of the indel would be, the probes that are specific to the region that is either gained or lost may not be there, but there are adjacent probes to the said region that might be altered due to the indel. So what ends up happening then is that it's what we would call near target capture, or some people refer to it as shadow capture. The adjacent probes would still capture the region specific to their particular complementary sequence, but as a result, due to the random fragmentation of the library, the adjacent sequence, which is the novel sequence of the indel, will be brought along for the ride. So the probe acts almost as a hook or as an anchor to then allow the adjacent novel sequence to come through. And then what you'll do is you'll sequence through the probe and then go into the novel region. And if you do not completely sequence through the novel region on one side, because we're doing paired end sequencing, you will probably get a majority of it on the opposite side. And because we do a pile-up sequencing, all of these unique reads are going to be piled up to the point where, depending upon how large the indel is, you'll get either partial or complete stitching of that due to the pile-up and compression of the read files. However, now to a comment of that, as we start to identify more information about these indels, and this is one of the advantages that we at Twist have always been able to promote because of other customers we work with, we can take that novel sequence and just make new probes. And then those regions might not necessarily be incompletely or insufficiently covered, but now we can make probes to them so that they're nice and uniformly covered in that new strain.
Okay, thank you, Mark. Another question that's asked is, what if every sample in the capture pool is negative? Will the capture still work?
So yes, yes, the capture will still work. And well, you will know that the capture still works because I'm actually going to show this slide here. So I get, to a certain degree, I guess maybe I was prepared for a similar question like this. So here's what ends up happening. In addition to having SARS-CoV-2 there, you are also going to be bringing along some human background. It's going to come along as you generate the double-stranded DNA, or as you bring along double-stranded DNA from the sample that has both the DNA and SARS-CoV-2, whether it's messenger RNA, some ribosomal RNA, or the single-strand RNA from the virus. And what we find out is that when you do a bioanalyzer trace of these post-captured libraries, you see that you have fluorescent units for the 10 to the 6, which far exceeds the other background fluorescence for the other samples, whether 10 to the 5, 10 to the 4, 10 to the 3, or human only. We are capturing some human because it's there to such a large extent. It is, I mean, when you think about it, this is where viral load and the ability for a method to capture something with small viral copies, it becomes immensely valuable. But we are seeing still human bleed through, so to speak, coming in with the capture. So this is confirmation that the hybridization is working. What we do not know in these samples is what amount of virus loading is there. And that's the reason why after hybridization and target enrichment, regardless of what the bioanalyzer traces look like, which they will look like this, you will still have to sequence it because you will not know if you see 0 viral copies, 1,000 viral copies, or 10 million viral copies. That would end up being embedded in the peak underneath this representative capture trace.
Thanks, Mark. Another question coming in. How many differences in each probe can be tolerated before it fails to capture? Does it depend on where these differences are in the probe sequence?
No, it does not. So this is one of the advantages of target enrichment. It's by definition, it's meant to be very tolerant of a lot of mismatches. Because now, for example, if you're going to have a mismatch which is greater than 70% and it's an absolutely contiguous region, the likelihood is that probe is not going to touch down. There's just not enough homology to drive it. It becomes very similar to doing, annealing of a PCR primer. However, these probes are made very long so that there's an awful lot of homology to allow single base pair or dinucleotide base pair mismatching that would occur internal to the probe. What I can comment on is that we have seen probes capture adjacent strands with only 80% sequencing homology. So for example, if you have a random fragment that was generated either mechanically or enzymatically, and you have a probe that's 120 base pairs and you need, let's say for example, 80% of that, which I think translates to about 95 base pairs, you can capture a fragment that only has 95 base pairs of homology, even though the probe is 120 base pairs. And those fragments that you're capturing usually have about a median fragment size of about 200 base pairs. So really, 110 to 150 base pairs of the fragment is flopping out in the wind. So there is an awful lot of toleration for having the probe be able to bind to not completely homologous sequence. That being said, if you have long strings of non-homology or even interdispersed strings of non-homologous regions, some of those probes are probably not going to touch down very well. Hence the reason why you may want to have an increased tiling of probes to sufficiently tolerate maybe some adjacent hyperpolymeric regions. And as we learn more about SARS-CoV-2, we can account for that with maybe spiking in new probes that are maybe stacked or tiled more so that you don't miss out on those hypervariable regions.
Thanks, Mark. Another question that came in is, so you mentioned in your presentation that we recommend 2x75 reads. I guess the question is why that particular size? Why not 2x150 for better assembly?
Okay, so the reason why we suggest 2x76 base pair reads is in our enzymatic fragmentation methodology, the median fragmentation size of our libraries comes out to be about 200 base pairs. Standard next generation sequencing likes to have the reads come very close to each other in the middle, but not quite touch, because the overlapping sequence doesn't really help us out too much. But also there's a Gaussian distribution of your libraries, which means you're going to have very small fragments, which will have an awful lot of overlap. And then there'll have on the other side of the curve, there's very large fragments where there's where there's less overlap. And the pileup files is how you fill the overlaps. The key with doing shorter sequences was to account for trying to minimize the excessive read overlapping because of the fragmentation insert size. But if someone wants to either generate larger fragments, or if they don't mind having more overlap in the middle, there's nothing preventing someone from actually doing that. The only thing to consider, there's actually two things that I would consider outright. One, the longer reads could result in more overlap in the middle on the median side if you have reads or if you have fragments that are, let's say, greater than or less than 200 base pairs. And secondly, it will increase the runtime for the sequencer, because the more bases you add to a read, the longer the sequencing run is going to be. So if all you're doing is generating overlapping reads in the middle of the insert, the question is it worth that time?
Okay. Here's a question. Can I use a different sequencing device like a BGI, MGI?
So that becomes, so here's what I would say with regard to our process. So our probes are agnostic to whatever sequencing methodology you want to use, because what they're doing is it's DNA capturing DNA. The key part to that is what adapter sequence you're adding to either side of your library. So on the library construction side, so the NEB side, which is the protoscript 2 and the non-directional second strand RNA synthesis, that's just generating double-stranded DNA so you can generate libraries. The library construction is composed of a fragmentation step, a polishing step, and a ligation step of the adapters with a UDI on it. The ligation step is specific to the adapter, which is specific to the methodology for sequencing. So if you can provide that adapter that's specific to your sequencer of choice, there should be no reason why you could not use this methodology. I mean, we're working with Illumina-based sequencers, but if you have a way to ligate an alternative sequencer's adapters, our probes are going to pull them down. The other thing you'll need is, in the hybridization, you'll need sufficient quantity and appropriate blockers to those adapters so that you don't get daisy chaining or concatenamers that are going to be generated during that process. There's nothing that prevents that. You just need to work a little bit on getting the correct adapters ligated in the library construction and making sure they're sufficiently blocked during the hybridization method. But our DNA will bind the complementary DNA sequences.
Thank you, Mark. Another question. Can you compare and contrast amplicon sequencing versus target enrichment sequencing, in particular the Paragon assay?
Yes. So the Paragon assay is an anchored multiplex PCR assay, and our methodology is a target enrichment assay. So in the amplicon-based assay, what you're going to do is, in essence, use primer pairs that will flank the region of interest, and then you're going to amplify the middle to generate the fragment that you will then take on to sequencing. So you end up having clonal amplification of the regions of interest, which will have the absolute same stops and the same starts. And those clonal amplified copies will then be utilized to do sequencing. With our hybridization-based methodology, what you're going to do is use complementary probes to hybridize to the seed strands of your target of interest, and then bring those molecules down, which are unique molecules because we don't overly amplify them, and then you'll sequence that. Now, there are pros and cons to both methods. The pros for a amplicon-based sequencing methodology is that relative to the amount of time that it takes to do a hybridization methodology, whether it be fast hyb taking a day to generate a sample or a 16-hour hyb taking probably 2 days to get to a sample, it is going to be shorter because you generate your sample in about 2 hours. You do the amplification for an amplicon-based sequencing run in about 2 hours, and then you're probably to a sequencer right then and there, and you have results either later that day or tomorrow. Even with our fast hybridization method, we're probably still looking at about a day and a half. The other advantage is that because it's a smaller panel, you're probably going to be able to exclusively avoid amplification or capture and amplification of non-specific or human-based regions because the primers are specific to those areas. So now let's say conversely to hybridization-based technology. Hybridization-based technology is going to be more advantageous in a situation where you want to diversify the panel of choice. So even though it might take us a little bit longer now to actually get to a result, it's going to be an awful lot easier for us to add additional content. And I'm talking about additional content that could be different probes specific to novel regions that we start to identify by monitoring mutation of the SARS-CoV-2 virus, or adding additional respiratory viruses, adding additional flu viruses, any additional kind of targets that you want to maybe expand the panel. So we don't necessarily have to be locked into one vector. We can go to a differentiation of disease states. Additionally, as we're seeing, and as I've shown with the SARS-CoV-2 genome, there's a number of mismatches that have been identified versus SARS that are localized throughout the SARS-CoV-2 genome. As SARS-CoV-2 starts to mutate at a rapid state, there could be additional single nucleotide mismatches that are going to be generated that could likely disrupt primer sites that are essential for amplifying regions that are specific to the SARS-CoV-2. That could result in that actually could result in a false negative, especially when you're doing surveillance and monitoring. Probes, though, because they are longer and they can tolerate those mismatches, are going to be less prone to that occurrence happening, which means that we're going to have less of an impact that I foresee moving forward with having fallout or false negatives that could be induced because of these mismatches having probes drop out. Additionally, we have adjacent probes that can act as a best redundancy to maybe capture near target regions and still allow some coverage to be made, even if a probe does not work appropriately. And finally, we can always add additional content in because our panels are going to be more adaptable to having additional content added, where you won't have to re-optimize a multiplex PCR and you wouldn't necessarily need to re-optimize the whole panel. You might just be able to run and go in less than 2 weeks. So I don't want to say that they don't both have their place, but maybe for like SARS-CoV-2, it's like using a hammer to put a nail in the wall. And maybe using expansive target enrichment panels that are hybridization based, it's like using a nail gun to build a house.
Thanks, Mark. One of the attendees had a question about pipelines for analysis. Do we have any recommendations, or what do we use? Venki, maybe this is something you can answer?
Yes, absolutely. So we have run all of our analysis that Mark just showed you through our internal pipelines. And we also have collaborated with a company called OneCodeX that is providing analysis free of cost, free of charge. So please go onto our website and you will find some more details around how to get onto the OneCodeX platform. You can directly use the output from your runs and upload them onto OneCodeX platform, and you should be able to run analysis for free.
Okay, thank you. We're getting close to 9:55, and I think we're running out of time here, so we want to be very respectful of everyone's schedule. For any questions that we didn't have the opportunity to address, please rest assured we will reach out to you directly and answer your questions in the next coming days by email. As a quick reminder, after you leave the webinar, you will be directed to a brief survey. If you have a minute to spare, we would really appreciate your feedback. Thanks again to both of our presenters, and thanks to all of you for joining us here today. Until next time, remember that science doesn't stop, and neither will we. Please stay safe. Have a good day.